Good morning, I'm Dr. Preeti Sharma. Let's talk on transamination and deamination reactions of amino acid. Transamination involves the transfer of amino group of alpha amino acid to alpha keto acid to form a new amino acid and a new keto acid. The enzyme that catalyzes these reactions are aminotransferase, and all transaminases require the presence of pyridoxal phosphate as in coenzyme. Pyridoxal phosphate is vitamin B6, active form of vitamin B6. So in this example, we will see the amino acid, one amino acid and one keto acid is there. And during the transamination reaction, transaminase enzyme in the presence of pyridoxal phosphate facilitate the transfer of this amino group to this keto acid. So result is the keto acid, this amino acid will convert into the keto acid and the keto acid will convert it up into amino acid. So resultant of this reaction is that we get from one type of amino acid to another type of amino acid and one type of keto acid to another type of keto acid. We can see one example, alanine along with the alpha ketoglutarate. Alpha ketoglutarate is actually it is a coenzyme of it is a keto acid of glutamate, and so alanine undergo transamination reaction. It gives this amino group to this alpha ketoglutarate. Alpha ketoglutarate in turn is converted up into the glutamate, and alanine converts up into the another keto acid, that is the pyruvate. Conjugate keto acid of alanine is pyruvate. Another example, this is the reaction facilitated by aspartate transaminase, and names to remember is very easy, is based on the name of substrate. Here we are using the aspartate for transamination reaction. So it is aspartate aminotransferase. Similarly, here it is the alanine undergo transamination reaction. So it is alanine aminotransferase. So names are very easy to remember. So in short, it is called AST, or the glutamate oxaloacetate transaminase. So aspartate along with the alpha ketoglutarate, in the presence of the enzyme and PLP, it converts up into oxaloacetate and glutamate. Here the transfer of amino group from here to this keto acid carbonyl carbon, and it becomes aminated and it converts up into the glutamate, while aspartate is converted up into the another keto acid, that is the oxaloacetate. So what we have seen in both the example, the alpha ketoglutarate is acting as an acceptor of amino group. So not only here, all the amino acids, whenever they undergo transamination reaction, so alpha ketoglutarate act as an acceptor of amino group, and all amino acids, amino groups are pooled into the glutamate. And glutamate is such an important amino acid. Here its amino group is derived from all the maximum amino acids which undergo transamination reaction. So it acts as an transporter and scavenger of this amino group. Otherwise, the released amino group from the amino acid as a ammonia will becomes very toxic, and to remove this toxicity, it is a way of transportation of ammonia via glutamate. So glutamate is funneling all the amino groups of all the amino acid into itself, and this way from all the peripheral sites the glutamate is collecting the amino group, ammonia as an amino group, and it is being taken to the liver. So very important reaction. Finally, this glutamate reaches into the liver, and where it undergoes deamination reaction. We will see subsequent. So in the previous slide, the net result is the formation of glutamate after the transfer of amino nitrogen to alpha ketoglutarate. So except for lysine, threonine, proline, and hydroxyproline, which is a derivative of proline, all these amino acids do not undergo transamination reaction. So these are the 4 amino acids which do not undergo transamination reaction, and rest of the amino acid their first relation reaction is transamination reaction. So there is no net loss of amino group in transamination reaction. Only it is just a transfer of from one amino acid to another. Now move on to the mechanism, how the enzyme along with the pyridoxal phosphate act as a transporter of this amino group of amino acids. So let's see the mechanism of how the amino group is transferred from amino acid to keto acid. What is the role of enzyme and the pyridoxal phosphate as a coenzyme? So initially what happens, this is the enzyme, it is the active site of enzyme, and there is a lysine amino group of the lysine amino acid which attacks onto the aldehydic carbon of the pyridoxal phosphate and form a kind of a Schiff base. And there is Schiff bases in which the doubly bonded carbon nitrogen bond is there. So this kind of a duct is known as Schiff base. In the next step, the amino acid amino group attacks onto this carbon, Schiff base carbon, and attaches itself to the Schiff base, replacing the lysine amino group. So this enzyme is detached now, and keto acid, after being hydrolyzed, it this entity separated as in keto acid, and this pyridoxal phosphate now converted into pyridoxamine phosphate because now amino group has been transferred from this amino acid. So the enzyme is also detached. In the next step, the amino group of the enzyme is now attacking onto the is now attacking here. It's not attacking, but this is the keto alpha ketoglutarate, and this amino group is now attacking onto this carbonyl carbon of the alpha ketoglutarate and forming this kind of product. Schiff base kind of bonding is there. So in the next step, this lysyl amino group of the enzyme, now it is attacking this carbon to replace this it is this amino group of the lysine is now attacking to this carbon of the pyridoxamine. So it is replacing this nitrogen, this amino nitrogen, and this whole entity is now separated as a glutamate. So this is how the transfer of amino group right from pyridoxamine to this alpha ketoglutarate, and it turns up into glutamate, and the enzyme complex along with the PLP is now separated. So this is the mechanism of how there is a transfer of amino group from one amino acid. This is the amino acid to another keto acid, that is always alpha ketoglutarate. So what is the biological and metabolic significance of transamination reaction? First important is like it leads to the breakdown of amino acid, and second is synthesis of non-essential amino acid. You have seen in the previous so many examples, like different kind of keto acids can be converted up into another kind of amino acid by simple transfer of amino group, and so this way we can both synthesize non-essential amino acids as well as keto acids, which are necessary in various metabolic reactions. Like pyruvate can be transaminated to synthesize alanine, oxaloacetate produces aspartic acid, and alpha ketoglutarate is transaminated to form glutamic acid. Those amino acids which cannot be synthesized in this manner are therefore essential, and they should be taken into the diet. And now third importance is the interconversion of amino acids. So by this way, one type of amino acid can be converted up into another type of amino acid, and this is a way of equalization of quantities of non-essential amino acids. Suppose one amino acid is in higher quantity and other one is less, so higher amount of amino acid will convert it up into the keto acid, and amino group will be transferred to a keto acid and leading to the increased concentration of the amino acid which was less in concentration. So this equalizes the quantities of. Now let's talk on to the clinically important transaminases. These are ALT and AST. ALT stands for alanine transaminase, and AST is aspartate transaminase. So these enzymes are important, clinically important in various liver and heart diseases. AST is increased in myocardial infarction, and ALT in exclusively in the liver diseases. And aspartate transferase and alanine aminotransferase are induced by glucocorticoid, which favors the gluconeogenesis. Deamination. This is a hybrid term. It's the combination of two terms, transamination and deamination reaction. So transamination, as we have seen in the previous slides, this reaction is most important reaction of amino acid, which is happening in all the tissues, and we have also seen that biological importance of transamination reaction. It is actually a way of transfer of amino group from all the amino acid to the glutamate, and from glutamate the glutamate reaches to the liver, and this is how it is funneling all the um, amino groups of all the amino acid to the liver. So the deamination, so glutamate comes to the liver, comes for a deamination reaction. So deamination maximally happens in the liver. So we can see here, this is any amino acid. It can be any amino acid. So during trans amino acid, the always another amino acid should be another keto acid should be a alpha ketoglutarate, because it will be acting as an acceptor of amino of the amino acid. So transamination reaction, which is happening in all the tissue, it converts up into the corresponding keto acid, and alpha ketoglutarate as usual will be converted up into the glutamate. Now glutamate is transferred from tissues to the liver. In the liver, the glutamate undergoes the oxidative deamination reaction, which is happen in the mitochondria of hepatic cells. So the enzyme glutamate dehydrogenase enzyme is available here only. And what is happened? It is an oxidative deamination. So NAD is molecular presence of NAD is required. NAD is converted up into the NADH. It squeezes out the hydrogens of this glutamate and removing the ammonia. So this is how the oxidation of glutamate to alpha ketoglutarate, and the ammonia is lost, and this ammonia will be converted up into the urea, and this happens into the mitochondria. Initial two steps of the urea synthesis happen to the mitochondria. So, and rest of the step happened in the cytoplasm. So this is how the urea synthesis will take place into the liver only, and alpha ketoglutarate is now regenerated, and it is all set to bring another slot of amino acids from various sites of the peripheral sites, and it is all involved in the transportation of amino group from peripheral sites to the liver. Now, deamination can be of various types. It can be oxidative deamination, and it can be non-oxidative deamination. And these deamination can be via L-amino oxidase enzymes also. So we'll see one by one. Oxidative and deamination reaction, we have seen it here. See, the glutamate dehydrogenase, we have seen in the previous slide how the glutamate dehydrogenase is converting the glutamic acid only, glutamate, into the alpha ketoglutarate, releasing the ammonia. And this is the reaction. This enzyme is present in the hepatic mitochondria, and alpha ketoglutarate is released, and ammonia is also detached right from glutamate, and this ammonia is derived actually the amino groups of all the amino acid. The second is non-oxidative deamination. So in this, there is no role of NAD or NADP. So serine, like for example, serine or threonine are there. So dehydratase enzyme is there. It also requires the presence of PLP. It converts them into the corresponding keto acid. Like for serine, it is a pyruvate and ammonia is lost. And for threonine, it is alpha ketobutyrate and ammonia is also released. So these are two ways, oxidative and non-oxidative ways of transamination reaction. Then L-amino oxidase, this is the enzyme which is evidently present in the liver and kidneys, and it is also involved in the deamination reaction. We can see in the subsequent slides. So this is amino acid oxidase. Amino acid is converted up into corresponding keto acid with the loss of ammonium ion in the presence of enzyme amino acid oxidase, and oxygen, molecular oxygen is converted up into H2O2. This hydrogen is being derived from this the oxidation reduction of FMNH2 system. So we can take example of glycine. Glycine is converted up into the glyoxylate, molecular oxygen converted up into the peroxide, and amino group as an ammonia. And FMN is converted up into the FMNH2. So this is the whole mechanism of reaction, how it happens, and how the amino acid oxidase is removing the ammonia.